fibrinogen into fibrin, activates brake pedals on itself by turning on antithrombin in the blood. But that's how heparin makes blood clotting prevent, right? How it helps to stop blood clotting. It increases antithrombin levels, right? Because if I asked you that, know that. That's how heparin from mast cells and basophils helps to prevent blood clotting. But let's finish with this. How do you get rid of the blood clot? Know this, because this was also in an assignment. Thrombin activates an enzyme called plasminogen, which is in your blood right now. But since it's got inogen after it, you know it's inactive. Plasminogen gets chopped by thrombin to become what? Plasmin, exactly. Plasmin, because plasmin is the enzyme that breaks apart fibrin. Plasmin is a chainsaw that can go up the fibrin in a blood clot and dissolve the blood clot. So if we give somebody tissue plasminogen activator, be clear, that's called a clot buster, but really what the clot buster is plasmin, right? Tissue plasminogen activator activates plasminogen in your blood to plasmin fast. So plasmin can then chew apart the blood clot to help somebody with a stroke or a heart attack get blood flow going again. Good. Yep. So that's a nice little blood 2 review. That stuff is definitely important to know for this exam, because this exam kicks it up a level from our first exam that had much less on it. So know those things for exam, and you've done a lot of work on those already. So let's jump into lymphoid part 1 review. And remember, with this review sheet, we first introduced who the pathogens are. Don't confuse a virus with a virus-infected cell. A virus is just a piece of DNA or RNA in a little protein shell called a capsid. It's not living, but it's got these little receptor proteins that it uses to dock up to human cells, infect the cell, and then make the cell make baby viruses for it, right? Use its ribosomes to make new proteins and new RNA, DNA, other ways to then make new viruses. Because remember, cytotoxic T cells love to kill virus-infected cells. Don't say they kill free viruses, they don't. Antibodies can bind to the free virus receptor proteins and neutralize them, but you can't kill the virus per se, right? Because it's not living. Bacteria are living cells, which again have receptor proteins that sometimes make us sick by themselves if they're in our blood or whatever. But sometimes the bacteria make protein toxins that can also infect cells and make us sick. That's like salmonella, right? The food poisoning from salmonella is not the bacterium that's really the problem. It's the toxin that it makes, okay? And even cancer cells have abnormal receptor proteins that a normal cell would not be making. They're dividing too fast, but they also make abnormal proteins. So all of these, if these are non-self cells and non-self things, they're all going to have antigens that are non-self. Remember, foreign antigens? And so we can have non-specific defenses against some of these things that don't care about what antigen, but then specific defenses mean the lymphocytes that are going after just one antigen, just one specific virus, influenza virus, coronavirus, the COVID virus, rhinovirus, right, cold virus, or they're going after one bacterial cell or one toxin. And that's why we get all those different vaccines to give us those harmless versions of the antigens so we can make memory lymphocytes to remember. We'll review all that as we go. So quick on lymph. What's lymph? Lymph is water that leaks out of your blood into the interstitial fluid and gets picked up by the lymphatic capillaries. Every day there's about 3 or 4 liters of water going into your lymphatic capillaries everywhere in the body. They're now called lymph. And we know lymph flows through the lymphatic vessels, goes through lymph nodes, goes into the spleen, goes into the thymus, right? Places where white blood cells are living that can help give us defense against different sort of pathogens. So remember, key details about lymphatic capillaries to remember, they're bigger and leakier than a normal capillary, right? That's important because with lymph, we see it coming up later. Those chylomicrons leave the small intestine in those big leaky lymphatic capillaries, the lacteals, the milky capillaries that leave the small intestine, but they're lymphatic capillaries. But every part of your body has lymphatic capillaries in it because if there's a blood capillary there, there's got to be a lymphatic capillary there to pick up that extra interstitial fluid, right? That extra water each day. But because they're bigger and leakier, even pathogens can get in or out of the lymphatic capillaries, into or out of the lymph, and even white blood cells can move in or out of the lymph. So here's what you got to remember. Most of the body's lymph, right, and lymphatic vessels, don't forget, also have valves, right? Valves and muscular compressions needed to squeeze the lymph back up to what duct mostly? What's the important duct? Thoracic, good, because remember the thoracic duct is right there where the left subclavian vein is. So all the upper left body, all the entire lower body, including the small intestine lymph, all dumps to the thoracic duct, which dumps into the left subclavian vein to dump all that lymph in. The right upper body uses a different duct to dump into the right subclavian vein, but that's the point is, this is even when you eat a meal that has a lot of grease, a lot of triglyceride in it, all your lymph from your small intestine also dumps into that same thoracic duct, okay? So lymphedema, y'all know before, is if the lymph vessels are messed up, lymph will accumulate in a limb, and not only could you get a swollen arm or a leg, whatever body part, but you're also at a higher risk of infection because now that lymph isn't leaving the body part well and getting to the lymph nodes where an immune response could trigger, okay? So go on, here. Every time I say poiesis, what's the poiesis mean? What's simple word for poiesis? Making, exactly. So when you hear poiesis, just go, okay, hemopoiesis makes any kind of blood cell. Lymphopoiesis simply means making lymphocytes. But it all goes back to the great granddaddy stem cell of them all, the hemocytoblast, good, which becomes either lymphoid stem cells or myeloid stem cells, right? You hear this every lecture. But then remember, for the lymphoid stem cells, they'll either become CD8, what? Cytotoxic T lymphocytes, or CD4, helper T lymphocytes, or B lymphocytes, good. And remember, cytotoxic T lymphocytes dock up to virus-infected cells or cancer cells or organ transplant cells and kill them. We'll review how they kill in the next lecture, but they're only going after only after one antigen on one specific target cell like that. Helper T cells also activate against one antigen, but remember, they don't kill things. They secrete cytokines, and we'll review what those cytokines do in the next review from the next lecture. B lymphocytes, when they see their antigen using their IgDs, they differentiate and become a plasma cell, heck yeah, plasma cell that secretes lots of immunoglobulins, good antibodies to go after that same antigen, because all this comes up a lot in the next lecture. We go into total detail about those. But remember, every kind of blood cell is always made in the bone marrow, but then the T lymphocytes go to mature and differentiate where? Thymus, that's what the T stands for. B lymphocytes do their maturing in the bone marrow, and all maturing means is differentiating thanks to having cytokines turning on certain genes in your DNA, right? Making you make certain proteins. Because if you got to mature, it basically means if I'm going to be a cytotoxic T cell, I got to know what antigen I'm supposed to go after and what I'm supposed to kill. I got to do that maturing in the thymus. But then when I'm done maturing, I leave the thymus and I can go to any place like the lymph nodes, the spleen, the blood, wherever I need to go. You only mature in bone marrow if you're a B lymphocyte and mature in the thymus if you're a T lymphocyte. Okay. So going on, remember lymphoid tissues that are not organs per se because they don't have a capsule. One are the MALT. What's MALT stand for? Mucosa associated lymphoid tissue. You've seen that a lot now, right? The MALT. There's two MALTs to remember. Peyer's patches, which are in the wall of your small intestine, especially in the ileum. And then intranasal vaccines that are given into your nose prime the MALT in your nasal cavity, not called Peyer's patches, but oral vaccines are used with the Peyer's patches, right? When you want to be resistant against maybe a virus that's going to come in from your food, oral vaccines get the Peyer's patches ready for that intestinal virus to be ready for it. Tonsils are also lymphoid tissues, right? You got palatine tonsils, which the palatine tonsils are the ones in your throat you can see, lingual tonsils under your tongue, and the pharyngeal tonsil called the adenoid. Those are all sitting in your pharynx area, ready for viruses, bacteria coming in through your pharynx, right through your mouth or your nose. But they're also called lymphoid tissues, right? And if you get your tonsils removed because they're always swelling up, not a big, huge deal, but it can be painful, right? It's a rough surgery because they're not really an organ. They have to kind of roto-root your whole pharynx to get those out. But lymphoid organs mean they simply have a capsule around them. And there's three: lymph nodes, spleen, and thymus, right? The lymph nodes are basically a mini kidney bean organ that's kind of like what you saw with the kidneys. Remember, there's a capsule, there's a subcapsular space, then deep to that is the cortex, and in the middle is called the medulla. And remember, who lives in each place? What two cells live in the subcapsular space? Got to know this. Macrophages and dendritic cells, because they are called antigen-presenting cells that get the lymph coming in from the afferent lymphatics and present antigens to what cells that live in the cortex? T lymphocytes, easy, because cortex has a T in it. And what cells that live in the medulla? B lymphocytes in the plasma cell. So everybody lives in a space. If it's in the review sheet, know that, right? And what's it called when you have swollen, painful lymph nodes? Lymphadenopathy, right? Lymphadenopathy, swollen, painful lymph nodes. And where do you tend to see those? Either in the cervical area for like strep throat, because there's a lot of lymph nodes in your neck area, in your axillary armpit area, because you've got an infection in your upper limb, you tend to see swollen axillary lymph nodes, or inguinal lymph nodes in your groin if you had an infection in the lower extremities, right? Because that's where you have the biggest number in most lymph nodes. But remember, each lymph node gets its own little blood supply to keep the cells alive, but it's got lymph dumping in and lymph dumping out. So it's kind of like perfect for looking at lymph to see if there's viruses, bacteria, toxins in it. Right? But cancer cells also like to metastasize some in the lymph, and they can set up camp in the lymph nodes, like for breast cancer. That's a big deal. So thymus, why do you go as a T lymphocyte to go to the thymus to mature? One, because you get your blood thymus barrier there, which means special capillary, basically epithelial cells guarding the capillaries, so that the immature T lymphocytes don't see antigens until they're full mature, right? Because you don't want to go against a self-antigen. So it kind of keeps you isolated as a T lymphocyte, having that blood thymus barrier. And number two, thymic what? Cytokines, good. The thymic cytokines or thymic hormones that are, again, the cytokines that T lymphocytes need to do their mitosis and their differentiation. And once they're fully mature and differentiated, you get to leave the thymus, right? It's only where you're mature. But don't forget, thymus too is also the organ that gets bigger till puberty. And then pretty much from your 20s on gets smaller through the rest of your life, right? It gets involutes. And it's the organ is, remember, right in the mediastinum, kind of like right here near your heart, kind of in front of that. So keep that in mind. And then number three, what's the organ that's made of red pulp and white pulp? You know it, the spleen, because the spleen is mostly red pulp. It's a fragile organ. It can easily be ruptured because it's mostly just loose tissue called red pulp, which are all sinusoidal capillaries, letting old red blood cells get phagocytosed by fixed macrophages there. And the iron can be stored in the spleen as ferritin or hemosiderin. You remember? It's another place in addition to the liver that you store iron as ferritin or hemosiderin. But the white pulp are the little islands of white blood cells living in the red pulp that have every cell there, macrophages, B lymphocytes, T lymphocytes, kind of looking for antigens in the blood, right? The spleen is more of a blood filter. Lymph nodes are more of a lymph filter, right? So if you don't have a spleen because you had a splenectomy done, What infections are you very risky prone to? Bacterial. Good. Bacterial infections are much more risky and much more serious for people who don't have spleens. That's an important one, especially bacterial pneumonia. So for non-specific defenses, keep this simple. Macrophages, neutrophils, right? They're sort of non-specific phagocytosers. I'll just highlight these because we did these in our reviews that you can go back to and look. Fever, pyrogens are chemicals that make the thermoregulatory center in the hypothalamus upregulate the set point. That helps white blood cells to activate at a higher temperature. Inflammation is all about mast cells and basophils secreting their histamine, their heparin, their prostaglandins, and that helps to get white blood cells to the area. But here, natural killer cells. Don't forget, one natural killer cell could kill a bacterial cell, a virus-infected cell, a cancer cell. They are highly versatile killers that are not specific for one antigen, but they still do cruise around the body all the time looking at antigens. If you're a non-self antigen cell, you're done, right? They can kill you pretty much. And the way they kill are those perforin proteins. Don't forget it. If you had to explain a perforin for a natural killer cell or a cytotoxic T lymphocyte that also use these, say they're a protein torpedo, a little protein pore that gets inserted into the cell membrane of the pathogen cell, lets water diffuse through the perforin and lead to lysis of the target cells. That's how they kill. You see perforins later with cytotoxic T cells. Complement, keep it simple. Complement is about 11 proteins floating in your blood right now, all made by the liver, that if they see an antibody hooked to an antigen, they start binding to that antibody in a cascade. One complement protein hooks to the next, and one, it can lead to that opsonization, right? Make it easier for macrophages and neutrophils to phagocytose. But number two, the complement proteins can make basically a perforin called the MAC attack, the membrane attack complex, that again lets water diffuse through that complement protein hole and lead to lysis of the cell. So it helps complements antibodies in getting rid of stuff. Okay, that's why they call it complement. And then physical barriers, skin, acid of your stomach, right? Antibodies and lysozyme in your tears and your sweat and your mucus. Those are sort of like out there in the front line trying to get rid of things. The mucociliary escalator in your lungs, the cilia trying to get crap out of your lungs, non-specific sort of physical barriers. But the specific defenses, remember this, right? When we talk about acquired or adaptive immunity, it means making memory lymphocytes that remember the antigen. But to do this, you actually have to be exposed to the antigen, right? To make these. So know these examples. If I say what's naturally acquired, it means nature doing nature thing. Active immunity means you met real viruses and real bacteria, got sick, but made memory lymphocytes from them because it's active. Your immune system did the work. Artificially induced active immunity means you still made your memory lymphocytes, you did the work, but you got a vaccine or a shot, right? A vaccination, that's artificially induced. Naturally acquired passive immunity means mom made the antibodies for you and they crossed the placenta as IgGs to protect you as a fetus, or they got into the breast milk in the colostrum to protect you as a newborn, right? But artificially induced passive immunity, you should know, would be like getting anti-rabies virus immunoglobulins because it's artificially induced. You had to get shots of those immunoglobulins at the doctors, right? But they were already made for you. Good. And then we move on to real important lymphoid part 2 review. And again, we did a full out review of this earlier, but let's make it quick and simple here to get to the point, right? Cytotoxic T lymphocytes, what do they do for us? Cell-mediated immunity, meaning they go up to other cells and they kill them. But let's first go, how do they recognize their antigen and activate against their antigen? This could be a nice little writing fill in the blank kind of question. It's easy if you practice exactly what we wrote out here, right? So first, what would 3 possible target cells be for a cytotoxic T cell? Virus-infected cell, or cancer cells, or organ transplant cells, right? Not all 3, but one of those with the right antigen. And if you had to go through the steps for activation, practice this, right? If this is fill in the blank, I'm going to use these exact kind of words in the fill in the blank. So first step, step 1 is the cytotoxic T cell has different T cell receptor proteins, including CD8, that dock up to MHC class 1 receptor proteins on one of these cells that's showing off a foreign antigen. Remember, MHC1s are being made in the cell. And if there's any foreign antigens, they get stuck in that little binding cleft of the MHC1 and shown off to the surface of the cell. It's like a mailbox saying, I got foreign antigens inside me, right? That's step 1. That receptor only matches to that certain antigen in the MHC1 receptor protein. But step 2 is that virus-infected cell will also secrete co-stimulatory cytokines that bind to another specific receptor protein on the cytotoxic T cell, and in a complex way, activate enzymes inside that lead to full activation of the cytotoxic T cell. I kind of made that simple, but practice it. That's a great question on a part 2, right, for this exam. And then after full activation, what do you make? Memory cytotoxic T cells that don't go all the way to the active killing stage, and then some active cytotoxic T cells that kill. If I say, how do they kill their target cell? Secreting perforin proteins that say make a protein hole in the membrane, leading to lysis of the target cell, and secreting cytokines that activate apoptosis, gene programmed cell death of the target. Those are 2 ways we covered that they kill. Maybe you got to explain those for the exam. But be able to do the same thing for a helper T lymphocyte. Go through the 2 steps for activation, but the difference is this is an antigen presenting cell like a macrophage showing off MHC class 2 receptor proteins that again have to do the same docking or matching to the receptor proteins of the helper T cell. And same deal, that macrophage will also be secreting co-stimulatory cytokines, the antigen presenting cell, that bind to another specific receptor on the helper T cell to activate enzymes inside for full activation. But again, same deal. We make memory helper T cells. We also make some active helper T cells that secrete cytokines that activate everybody. Helper T cells, cytotoxic T cells, B cells, macrophages, natural killer cells. And that's why HIV, good question, how does it cripple your immune system? It kills the helper T cells. And without the cytokines, you don't stimulate all those different 5 cells that we wrote out. Your whole immune system starts to fall apart. And then for B lymphocytes, remember how they activate against their antigen. They don't have receptor proteins per se. They have IG, what? Ds, good. They use immunoglobulin Ds as their antigen receptor, which are antibodies that can bind up to or dock up to an MHC class II receptor protein on a helper T cell that's showing off that foreign antigen. But same deal, the helper T cell has to secrete co-stimulatory cytokines that bind to another receptor on the B cell to activate enzymes inside that lead to full activation, because we did a nice full out review of this back with Lymph2. And then after full activation, remember, you make some memory B cells and also active B lymphocytes, which are called plasma cells, that secrete a ton of antibodies, right, against whatever that antigen was. And then if you flip to page 4, don't forget the difference. MHC1s are made by all cells all the time. They're also called human leukocyte antigens, because for people that are going to get an organ transplant, they look at the antigens in MHC1 for tissue typing to see how close you are for the match. But MHC2s are only made by antigen-presenting cells, macrophages, dendritic cells, and on helper T lymphocytes to show off those antigens. And then for antibodies, what you got to remember about antibodies, 80% are IgGs. They're the small ones that are great at neutralizing viruses and bacteria. They're the ones you make in your second exposure to the antigen mostly. And anti-RHs are IgGs. They can cross the placenta. IgAs are the antibodies of secretions. So think mucus, tears, saliva have IgAs in them. IgM is big old macroglobulin, 5 antibodies hooked together. Anti-A and anti-B antibodies are IgMs. And they're also the antibody you make in the first exposure to the antigen, right? The first time you see the pathogen, it's not as good at neutralizing viruses and bacteria, but it's amazing at agglutinating red blood cells and causing those massive transfusion reactions if you got the wrong blood type, okay? It can't cross the placenta, it's too big. So a mom could definitely have a B blood type baby and she's A. It's not like it crosses the placenta, but if you got the wrong blood, it's gonna cause massive agglutination, massive hemolysis. What's the Ig that's on the surface of mast cells and basophils? IgE, right? Allergies, parasites, remember? And then IgD is the immunoglobulin on the surface of B cells, right? Looking for antigens. And then here, remember antibodies, when we talk about the primary immune response, if I show you that graph from the notes and you see IgM going up and it takes a week and a half for IgM to go up, that's the first time you see the antigen. If you see the antigen the second time because you've already been vaccinated against it, you'd mostly see IgGs going up and they go up wickedly fast in just a couple days, right? That's the secondary immune response. Go back to that review if that sounds foreign. And then how do we get rid of stuff with antibodies? Either, neutralization or triggering opsonization, making the complement cascade trigger with antibodies, or agglutination of cells, or again, mast cells and basophils by having IgEs on them to trigger their degranulation. It's another way to help to trigger inflammation, okay? So that's our big lymphoid part 2. And then going on to digestive, hey, we just finished reviewing all of this, our last couple lectures. You should definitely be able to look at a picture from the lab manual or, from your lecture textbook, but we did this especially with nice lab handouts. Know your order of layers, because even for lecture, so it was part of lecture, this could be a good question to know about the mucosa, the submucosa, the inner circular, the outer longitudinal layers of muscularis externa, all that smooth muscle, the serosa, the visceral peritoneum, right? That's all fair game for lab and for lecture. But then remember the nerve plexuses that are built into the wall. The submucosal plexus has a lot of sensory nerves and pH nerves to detect when food comes in, but it's also the plexus that makes glands do their secretions a lot. The myenteric plexus of nerves are mostly parasympathetic nerves that are helping to trigger the smooth muscle of that muscularis externa to do peristalsis or churning, right? And then we'll kind of highlight just a few things here as we talk about nerves, hormones, and local in a moment. But don't forget the tongue, innervated by cranial nerve 12, the hypoglossal nerve, is mostly muscle. That's how it moves the tongue around, hypoglossal nerve. But it makes the enzyme called lingual what? Lingual lipase, good. Because lipase, don't forget, digests triglyceride into 3 fatty acids plus glycerol, good. The salivary glands, they make alpha amylase. Remember, amylase digests what? Starch into glucose. Got to know that, right? And there's also some IgA and lysozyme, the enzyme that breaks bacterial cell walls, in your saliva as well. I'm highlighting only a few things quick because we can go back to these reviews for all of this. The swallowing reflex, don't forget, starts with the buccal phase. That's when you can spit the food out. It's voluntary. But then once the food gets back into your oral pharynx, it's now the pharyngeal phase. Pharyngeal phase and esophageal phase are all cranial nerve 9 and 10, bringing sensory to the medulla swallowing center. Remember, swallowing center in medulla? It's all involuntary. And then cranial nerve 10, the vagus nerve, sends motor commands back to the palate, the epiglottis, to do all the things it needs to do to swallow the food down the esophagus. And then remember, once the food is down in the esophagus, peristalsis pushes it into the stomach. So you should definitely know your anatomy of the stomach. We did that in both lab and in lecture, right? Know the different layers, know the rugae, the function, the extra oblique layer, right? The pyloric sphincter, you got to know all that to have any of the physiology. But then if we flip, remember stomach gland cells. What are the cells that secrete acid? Parietal cells. You have to know this. There's going to be questions about the GI hormones that test you on this. And then they make intrinsic factor as well for vitamin B12 absorption. What are the cells that make pepsinogen? Chief, good, because chief cells secrete pepsinogen, inactive protease that gets activated by hydrochloric acid to become pepsin, to then start chopping proteins to amino acids. That's the first protease, pepsinogen. Not until you get to the stomach, you get a protease. Now remember, pylorus makes a lot of mucus to protect the stomach, but Helicobacter pylori is the bacterium that can mess up the mucus layer to cause what? Ulcers. Good, that's the bacterium linked to ulcers. And then we'll review gastrin, all the 4 GI hormones on page 9 to get to the point. But remember, for gastric activity, there's 3 phases to gastric activity. Stomach activity, one's the cephalic phase. What's cephalic mean? So it makes sense. Head means head. So when you're dreaming of food, smelling food, seeing food, and you're not actually eating it, cephalic phase means vagus nerve sends parasympathetic commands down to get all those cells secreting, and also to get the muscularis externa even churning, thanks to making a lot of gastrin, right? But even tells the pancreas, start making digestive enzymes back in the cephalic phase. Gastric phase is all that neural response, the hormonal response by gastrin, just making those same cells do more of their secretions and more of muscularis externa, smooth muscle, doing the churning. Intestinal phase, what you should remember is when finally the pyloric sphincter opens up and the food, the chyme, dumps into the duodenum, there's the enterogastric reflex. The small intestine tells the stomach, contract the pyloric sphincter, stop all the churning and, the contractions of muscularis externa, right? Stop gastrin from being secreted and start secreting more mucus in the duodenum to protect from all that chyme. But it's also from the duodenum where we see the next 3 GI hormones, secretin, cholecystokinin, and vasoactive intestinal peptide come out of the duodenum, enteroendocrine cells, to then go in the blood to do different effects, right, in different organs. So if we flip to small intestine, don't forget duodenum, jejunum, ileum, of course, we did this in lab and in lecture. All the absorptive surface area increasers, right? You should know plicae circularis, villi, microvilli for lab and lecture of how the jejunum has all that to increase surface area for nutrient absorption, like glucose and amino acids, right? But remember, triglyceride has to leave in the chylomicrons to go into the lacteales, the leakier lymphatic capillaries, to eventually get back to your thoracic duct to go into your blood. But that's also where the fat-soluble vitamins A, D, E, and K get absorbed, or in those chylomicrons. And then brush border enzymes, the one big one to remember is trypsinogen that sits on the microvilli of the small intestine, enteropeptidase, which chops trypsinogen into trypsin, the active protease, because when we talk about pancreas next, don't forget pancreas, right? Its big jobs, we'll make this quick, are to secrete digestive enzymes made by the pancreatic acinar cells, and also alkaline bicarbonate-rich pH buffer to neutralize the chyme coming in. And you should definitely know all about hepatopancreatic sphincter, better known as the sphincter of Oddi, and the ampulla of Vater, how that regulates when bile and pancreatic juice can dump into the duodenum, right? Because we talk all about that when we talk about GI hormones. So don't forget effects of secretin and cholecystokinin on the pancreas, and also review all your pancreatic digestive enzymes that digest everything that we eat. Well, I'll review all the digestive hormones in a moment when we get to the page 9. Liver, I'm going to say go back and review anatomy of liver. Hepatic portal vein brings all that nutrient-rich blood to the liver. Hepatic artery just brings the arterial blood to the liver. Know your portal areas, right? What 3 vessels are found in the portal area? A branch of the hepatic artery, a branch of the hepatic artery, a branch of the hepatic portal vein, and a branch of the bile duct, right? Because that's how they get bile out of the liver. Kupffer cells are those fixed macrophages. Review the jobs of the liver, right? About insulin, helping the liver to make glycogen, make proteins, and make triglyceride, and how glucagon helps to break those things apart when you're not eating, right, in the liver. How the liver stores iron as ferritin and hemosiderin, fat-soluble vitamins A, D, E, and K, all stored in liver, vitamin B12 stored in liver. Liver cells do a lot of drug and toxin metabolism. We know that. The fixed macrophages in the liver do what they do. Storing toxins and heavy metals is another job that the liver can do for us to protect the rest of the body, making 90% of the what? Plasma proteins, right? In a couple lectures, and definitely making bile, because bile's got bilirubin in it, but it's also got bile salts made from cholesterol that gets stored in the gallbladder, the cholecyst, to be concentrated. And then we got into CCK and all of its big effects, because CCK, remember, its name comes from contraction of the smooth muscle of gallbladder wall. But remember, if we just flip to page 9 for a minute, review this. This is a great question for part 2 on this exam. Know this page, because it's my one summary of everything to know about the 4 GI hormones. First, gastrin is made by G cells, enteroendocrine cells in the stomach, mostly the pylorus in the duodenum, but mostly in the pylorus of the stomach. And it stimulates increased hydrochloric acid secretion by parietal cells, increases pepsinogen secretion by chief cells, increases contraction of smooth muscle in muscularis externa for more churning. These will definitely be easy fill-in-the-blank questions. Practice them, study them, because I'm going to ask you these for sure, because it reviews the whole digestive system with this one question. Cholecystokinin, secretin, and VIP are all made and secreted by duodenum enteroendocrine cells when chyme arrives. And CCK, remember its name is contraction of smooth muscle in the wall of the gallbladder to squeeze the bile out, but cause relaxation of the smooth muscle in the sphincter of Oddi to let the bile go into the duodenum, increases secretion of digestive enzymes from the pancreas, increases secretion of bile from the liver. It's a test to just see that you know what each organ makes. And they all, secretin and cholecystokinin, inhibit everything we just said that gastrin does for the parietal cells, the chief cells, and muscularis externa. Secretin also tells the pancreas secrete more alkaline bicarbonate-rich pH buffer, not digestive enzymes, and stimulates liver to secrete more bile, just like CCK. And vasoactive intestinal peptide, don't forget it's two different effects, and tell me why. It increases secretion of mucus in duodenum. Why? What for? Protect the mucosa of duodenum from the hydrochloric acid, from the chyme coming in, and stimulates vasodilation of capillaries in small intestine. Why? To get more blood flow, to absorb more nutrients, right? Cool, because that to me tests you the whole digestive system in one question. It really summarizes everything that gallbladder does, pancreas does, right? Liver, everything, right? The stomach. And then large intestine, to make this quick and simple at the end, mostly for water reabsorption. About 1.5 liters of water comes in when you fill the colon, and about 200 mils go out when you have feces, right? That's how all that water gets reabsorbed. And remember, parts you should know for lab and lecture, the anatomy is easy, but then a lot of mucus cells to lubricate the feces, and a lot of bacteria live in your colon. That's called the colonic microbiome. And they do different jobs, making vitamin K, converting bilirubin to urobilins and stercobilins, converting leftover foods that we didn't digest into gases like indole, scatol, and hydrogen sulfide. Good bonus points, right? Those are the 3 gases that make poop smell and make fart smell, right? And right, a lot of research into the bacteria in terms of how your immune system develops, obesity, right? Things that we didn't even realize this microbiome, your colon, is responsible for, right? What bacteria you have living there. And this is good to know for another little possible part 2 question. What's the defecation reflex? How does it happen? And it's really simple if you look at the review sheet. This trigger is when your rectum gets stretched, right? When the rectum gets stretched by feces stretching it, it triggers 2 reflexes. One is a short reflex just in the wall of the rectum locally. Another is a long reflex going back to your spinal cord. So the short reflex is simply peristalsis in the rectum gets triggered that leads to rectum pushing feces down into the anus. Simple. It doesn't need the spinal cord. The long reflex circle is when the spinal cord gets involved. The spinal cord is getting stretched from the rectum sent to it. And the spinal cord does 2 things, well, 3 things. One, it does a long-range peristalsis called a mass movement, which simply means peristalsis down the descending colon and the sigmoid colon. Know that, to push more feces down into the rectum to the anus. And then 2, relax what sphincter? Got to know that. Good. Relax the internal anal sphincter made of smooth muscle, which is involuntary, and automatically contract the external anal sphincter, which is skeletal muscle and is voluntary, because otherwise you wouldn't have bowel control. That's when you can then relax that external anal sphincter when it's time to have your bowel move. So know that stuff for this exam, and you will rock out. So go back and look at any of those reviews that you might have forgot things for, and this should be the best exam of all, with the most study time of all.